Hello everybody. Good morning. Hope you all are quite okay and fine. Welcome you all to my today online class. Before going to start my today's lecture, first of all, I would like to introduce myself to all of you. My name is Mohammad Ghulam Morshed, lecturer of biology, Milestone College, Uttara, Dhaka. Welcome you all to my today online class. Today I am going to discuss about chapter 10, that is defense system of human body, immunity. The topics today I am going to discuss about the structure of the antibody. First of all, I will be discussed here the types of antibody. I will be discussed here the functions of the antibody. These are the topics that will be discussed here today. Learning objectives. After completing my today's lesson, student would able to define some important definition like antibody, antigens, paratopes, hapten, and epitopes. You can also explain the different types of antibody. You can also explain the functions of the antibody. Now, first of all, we have to know about the antigen. What is antigen? Antigen, there are two words. Actually, anti means antibody and gen means generator. So, antigens is called the antibody generator. Look at the figure, look at the a very simple definition here. The substance who is recognized as foreign substance that when introduced into the body is capable of stimulating an immune response is called the antigen. So we can easily say that the antigen must be foreign to the host or any foreign substance that when introduced into our body will stimulate the formation of antibody or immune response is actually called the antigen. Antigens includes parts, cores, capsule, cell wall, flagella, fimbriae, and toxins of the bacteria, virus, and other microorganisms. But sometimes some non-microbial agents can also act as antigenic. For example, insect venom, pollen grain, egg white, these are also act as an antigenic agent. These are the non-microbial antigenic agent. Now the definition of epitopes and hapten. These two definition you have to know about this. These two definition. First of all, I would like to discuss here the epitopes. Epitopes, the part of the antigen proteins that is actually recognized by the immune response, specifically by antibody, B cells or T cells, is known as antigenic determinant or epitopes. Actually, epitope is the part of the antigen that actually recognized by the antibody, B cells or T cells. Haptens, some chemical substance that when react with a specific antibody but cannot induce the formation of antibody unless bound to a special carrier proteins or other large antigenic molecules, these are called haptens. Basically, haptens are very smaller molecular weight, have a very smaller molecular weight. Most of the lipids are actually act as antigen haptens, but when they cannot induce the immune response, but when they combine with some carrier protein, they act as antigenic agents. These types of low molecular weight of lipid is actually called the haptens. Now the definition of antibody. Antibody is any of a large number of protein of higher molecular weight that are produced normally by specialized B cells after stimulation by antigen and act specifically against the antigens in an immune response. So very simply, we can say that the antibody is a chemical substance that actually produced by the plasma cell in response to the antigen actually. The the production of the antibody is actually produced by the activated B lymphocyte. As you know that the B lymphocyte is actually originated from the bone marrow, but maturation of the B lymphocyte is also take places in the bone marrow. But when the B lymphocyte become matured, a specific receptor protein will be developed, and when the specific antigen binds to the receptor protein of the B lymphocyte, the B lymphocyte become activated and undergoes repeatedly mitotic division to produce numerous clone B lymphocytes. Some of the B lymphocytes are differentiated into plasma cells, which is responsible for production of antibody, and some of the B lymphocytes are differentiated into memory cell, and the memory cell is more active when this same antigen is enter into our body. These memory cells become activate uh, rapidly to produce numerous activated B lymphocytes, and some are differentiated into plasma cells, and some are again differentiated into memory cell. Definition of antibody. Antibodies are actually large chemical molecules of glycoprotein which is secreted from the B lymphocytes or plasma cells or the B cells or plasma cells. Human body able to produce 100 million types of antibody. Now the differences between the antigen and antibody. Antigen and antibody. If you have a clear understanding about the antigen and antibody, then you can easily differentiate between the antigen and antibody clearly. Now the antigen. An antigens is a foreign substance that causes the immune systems to produce antibody against it. An antibody is a protein that is produced by immune system when it detects harmful substance to neutralize or fight it. Antigens are made up of either polysaccharides or proteins or lipid. Antibodies are made of organic structural protein unit. Antigens can be cells, but antibody are never cell. Antigens act as a key, but antibody act as a lock. Antigens are typically divided into self and non-self antigen, but there are total of 5 types of antibodies like IgA, IgE, IgG, and IgM and IgD. Now let's discuss about the structure of the antibody. This is very important topics. You have to have a clear understanding regarding the structure of the antibody. Look at the figure here. This is the structure of the antibody. Antibody is a Y-shaped structure. Now I would like to discuss in details about the structure of the antibody. Each antibody is an immunoglobulin Ig monomer. The Ig monomer is a Y-shaped molecule that consists of 4 polypeptide chains, 2 identical large heavy chains and 2 identical small light chains are actually connected by disulfide bond. Each chain is actually composed of a structural domain called immunoglobulin domain. Heavy chains with one variable domain followed by a constant domain, a Hence region, and the 2 more constant regions are also found. Light chains with one variable and one constant domain. The light chains contain about 200 amino acids, whereas long heavy chains contains about 440 amino acids. The rod-shaped base of the Y-shaped antibodies consists only constant domain of heavy chains. This region is actually called the fragments crystallizable region or FC region. Each of the 2 expanded arm of the Y-shaped antibodies consists of both heavy and light chain. This region is called the fragments antigen bindings region or FAB region, FAB region, which contains both variable and constant domain of each polypeptide chain. The variable includes the end of the light and heavy chains, which is the antigen binding site. The antigen binding sites of an antibody is known as the paratoph. Due to recombination of the amino acid in the paratoph through a variable diversity joining, there will be 100 million types of antibody produced in our body. Types of antibody. Now I would like to discuss about the type of antibody. In human body, in human blood serum, there are 5 types of antibodies or immunoglobulin molecules. The type of antibody generally determines by the sequence of the amino acid in heavy chains of the antibody. There are 5 types of heavy chains in human antibody based on the sequence of the amino acid. These are the gamma chains, mu chains, alpha chains, epsilon chain, and the delta chain. Accordingly, the antibody of the human immune systems are classified into following classes. Now, number 1, the immunoglobulin G or IgG. This is a general type of antibody with gamma chain. This is the most abundant classes of antibody in the body and constitute approximately 70% of the total and is plenty in blood and lymph. It is a kind of antibody that works efficiently to coat microbes, speeding their uptake by other cells of the immune system. These classes of antibody can only transfer from mother to fetus through immunoglobulin M or IgM. IgM antibody with mu chains and remain attached to B cell. IgM constitute about 5 to 10% of the all antibody in blood. It is very effective in killing the virus and bacteria. It is the largest Ig, which is first secreted by the plasma cell. Now the immunoglobulin A or IgA, it is an antibody with alpha chain concentrated in the body fluid, tears, saliva, and secretion of the respiratory and digestive tract, guarding the entrance of the microbes to the body. It is the second most abundant classes, constituting about 10 to 15% of the antibody of the serum. If IgA found in the colostrum with huge amount. Now the immunoglobulin E, it is a rare type of antibody with epsilon chain and constitute about 0.1% of the total antibody in blood. These types of antibody is actually located on mast cell and the basophil. It influences releasing histamine, which active the response of the inflammation. It is involved in allergic and hypersensitivity reaction and provide protection against parasite. Now the immunoglobulin G or IgD. It is an antibody with delta chain and constituting about 0.2% of the all antibody in blood. These types of antibody found in blood, lymph and B cells and plays a key role in initiating early B cell response. Now look at the figure here. This is the IgG. IgG is a monomeric form. IgE is also monomeric antibody. IgD is also monomeric. But in case of IgM, these are the pentameric, and in case of IgA, these are the monomeric and dimeric, and sometimes they also trimeric form. Now the functions of the antibody-antigen reaction. A reaction that occurs when an antigen combined with the corresponding antibody to produce an immune complex is known as the antigen antibody reaction. Now, the functions of the antibody antigen reaction. Each antibody has a unique binding site called the paratopes, which lock onto the specific shape called epitopes of antigen. Antibody destroy the antigen, which is then engulfed and digested by macrophage. Now, antigen antibody reactions are on following types: agglutination, aggregation of microbes or cells due to the antibody bindings is known as the agglutination. IgM is responsible for agglutination antibody. Opsonization. The antibody that is actually making the antigen susceptible for phagocytic is called the opsonin. Activation of complement system. Antibody binding with antigen is not sufficient to remove the antigens from the body. Then antigen antibody complex initiate the activation and bindings of the complement systems to it. This is called the complement fixation. Lysis. Complement activation can result in the lysis of the bacteria. Thus, complement fixing antibody IgM may indirectly causes lysis of the susceptible target. Neutralization. Neutralization refers to the ability of antibody to block the site on bacteria or virus that they use to enter their target cell. Now the homework. Write down the functions of the antibody. Creative questions. Dr. Tufajul said there is a Y-shaped protein substance in human body. He added that protein substance keeps the body free from the certain disease. A. What is antibody? What is meant by paratope? Explain the structure of the mentioned substance. Analyze the last sentence of the Dr. Tufajul. Thank you very much for your passion sharing. I think today's lecture was very much effective lecture for all of us. I am humbly requesting you all to watch my video lecture for several times so that you can understand each and every topics that have already discussed here today. If you have any queries regarding my today's lecture, feel free to ask me, or you can also ask your respective subject teacher regarding your query. Thank you everybody. Thank you all.